All right, everyone, welcome back to our final lecture presentation, which will cover Chapter 29. Chapter 29 will cover the concepts behind pregnancy, development, and inheritance, and in terms of the last chapter, we're basically going to cover everything after the moment of fertilization. We discussed that in Chapter 28 with the reproductive systems, but here we're going to move from fertilization through implantation, development, and pregnancy, as well as some concepts behind how we inherit traits. We are building on the reproductive systems, and we have to keep in mind that fertilization took place in the female reproductive system, and implantation is going to take place there as well. So all of the events that we're going to discuss in this chapter can be localized to the female reproductive system. Now, we are all aware that pregnancy lasts for right around 9 months. To be even more specific, we would say about 38 weeks. However, during pregnancy, development is taking place, and we more accurately depict the events that are taking place during development in different subperiods within that 9 months. There's two major periods that we see throughout the process of development, the embryonic period as well as the fetal period. We're going to discuss the embryonic period first, as it occurs first. The actual parameters of the embryonic period are from the moment of fertilization all the way through the 8th week of development, or we could say the 2nd month. It is for this 8 weeks that the developing entity is known as an embryo, and it is after these 8 weeks that we're going to enter into the fetal period, where we will then call the developing entity a fetus. The fetal period lasts from the beginning of the 9th week all the way to the end of the 9th month. And we see different developmental processes taking place in the embryonic period and the fetal period. So picking up from our last chapter, we're going to start at the concept of fertilization and start making our way through the embryonic period. So we did discuss in the last chapter that fertilization is when a sperm cell penetrates a secondary oocyte. This is going to complete meiosis II and produce an ovum, and for a very short time, the ovum is going to have a separate male pronucleus and female pronucleus. Eventually, those two pronuclei are going to fuse, and that's going to form the zygote. And it is the zygote that contains a new set of DNA that has never existed before. Now, these concepts were covered in our last chapter, but I will just mention that during sexual intercourse, sperm cells are deposited in the vagina. These sperm cells are going to make their way through the cervix, up the uterus, and eventually in the uterine tubes. And it is going to be in the fallopian tubes where fertilization is generally going to take place. We must remember that in order for a sperm cell to penetrate the oocyte, it has to utilize the acrosome that's found on its head. The acrosome is going to contain digestive enzymes that are going to help it make its way through the corona radiata as well as the zona pellucida. The first sperm cell that penetrates the oocyte is going to be the one that fertilizes the egg. If you're anything like me, you might wonder how the sperm cells find the oocyte in the first place, since they're so incredibly small. Well, there's a special receptor on the zona pellucida known as the ZP3 receptor, and this is going to be the receptor for the membrane proteins that are found in the head of the sperm cell. When the sperm cell makes it to those receptors, the acrosomal enzymes are going to come in contact with the zona pellucida, digesting this layer and allowing the sperm cell to make its way in. The flagella is also going to be very important for this, as that's going to help the sperm cell push its way into the oocyte. You can see the ovum shown right here on the left side of this slide, and we can actually even see the male and female pronucleus that are getting ready to fuse. Once these come together, that's going to form the new diploid zygote, and then that zygote is immediately going to start the process of development. Once fertilization takes place, the zygote is going to undergo lots and lots of changes. But right around a day after fertilization has taken place, the zygote is going to begin the first stage of mitotic cleavage. This is just a fancy word for cell division. But the zygote is going to undergo a very specialized type cell division that occurs very, very rapidly. This first cell division takes about 6 hours. But each cell division after this is going to take less time than that. So roughly we could say that at the end of the 30th hour after fertilization, the first mitotic cleavage has come to an end. You can see over here that this is the 2-cell stage of the zygote. The zygote is going to have various different stages as it's constantly going to be developing. It started as one cell in what we would say the 1-cell stage. But eventually the zygote is going to undergo its first cell division and form 2 blastomeres. These are just simply the names of the cells that are produced from the mitotic cleavages. Blastomeres are going to be half the size of the parent cell that produced them. So you can also see the zona pellucida shown right here, and this is not going to be getting any bigger. In fact, all that's happened in here is that these cells have gotten smaller. And every single cell division is going to act the same way. The zona pellucida stays exactly the same size, but it's the cells themselves that are decreasing in their size. Cleavage of the zygote is going to continue into the 4-cell stage, where we'll have 4 blastomeres, and you can see that these are again much smaller than the 2-cell stage blastomeres. Around the 2nd day after fertilization, the 4-cell stage of the zygote is going to be present. Keep in mind that it took 1 day to start the first cell division, and then that first cell division took 6 hours. That puts us at 30 hours, but each cell division is going to be shorter than 6 hours. So therefore, this second cell division would be completed sometime during that 2nd day. We're going to have subsequent cell divisions into the 8-cell stage and eventually into the 16-cell stage right around the 3rd day. Still, even at this stage, the zona pellucida has not gotten any larger, and this 16-cell stage zygote is going to still be the same size as the 1-cell stage zygote. So to continue with our understanding, once the 16-cell stage zygote undergoes its cleavage, that's going to produce the 32-cell morula. Morula stands for mulberry, and that's exactly what it's going to look like here during day 4. You'll see the morula is still wrapped up in its zona pellucida, and the blastomeres are just continuing to get smaller and smaller and smaller. But this is going to be the first stage where we're no longer calling it a zygote. Here at day 4, we finally have the morula, and in order for us to have the morula, we have to be in the 32-cell stage. Now, I did forget to mention this just a second ago, so I will just note that the 16-cell zygote is actually shown here at the head of a pin. You'll remember that this is exactly the same size as the 1-cell stage zygote, and that's going to be one of the largest cells in the entire human body. Now, we've made our way to day 4, and we're in the current stage of the 32-cell morula. By this point, on day 4 or 5, the morula is going to finally enter into the uterine cavity. Now, the uterine cavity specifically is lined by the endometrium, as we should remember from our last chapter. The endometrium is going to contain glands that are secreting certain substances. These substances specifically are very glycogen-rich. These substances are going to leak into the morula and accumulate inside of this entity. The presence of the fluid is going to cause the morula to rearrange itself and form a large central cavity known as the blastocell, also known as the blastocyst cavity. It is at this point that we're now going to call the morula the blastocyst. And you can see a section blastocyst shown right here. This is going to be the rearranged morula where we have this blastocell right here, and then 2 different cell populations that we're going to be able to find, the embryoblast as well as the trophoblast. I've highlighted the embryoblast shown right in here, and the trophoblast is going to be found towards the outside. As the name suggests, the embryoblast is going to give rise to the embryo, and the trophoblast is going to give rise to part of the placenta. So between day 4 and day 5, we're going to have the development of the blastocyst, and this is going to be the entity that's prepared to implant into the endometrial lining. The blastocyst will generally hang out inside of the uterine cavity for a couple days, so right around day 6 is generally when implantation takes place. Just bear in mind, we are using fertilization to start off our timeline. So it is 6 days after fertilization that the blastocyst is going to implant into the uterine wall. Now, generally, we will find the blastocyst implanting into the body of the uterus or perhaps into the fundus of the uterus. But regardless of that location, what's always going to be the same is that the embryoblast will orient itself towards the lining of the endometrium. The embryoblast has to be oriented towards the endometrium right around day 6. So to summarize these events that have taken place so far, the ovary has at this point ovulated an egg cell, and the oocyte is making its way up the fallopian tubes. At the same time, sperm cells have been deposited, and they've made their way up the uterine cavity and into the uterine tubes. So it's up here inside of the fallopian tubes that we're going to have fertilization. This is generally going to occur within the first 24 hours after ovulation, and there is a very narrow window for fertilization to take place. There's a little bit of leeway as sperm cells can survive in the female reproductive system for several days. However, after ovulation, the sperm cells will need to make their way to the egg cell for fertilization very soon after. Now, once fertilization has taken place, we're going to have the fusion of the pronuclei and eventually form the zygote. And about 24 hours later, we're going to begin that first cell division. The first cell division is going to take about 6 hours, which is where we get this number, 30 hours after fertilization. That first cleavage is finished 30 hours after fertilization. Now, after this, each subsequent division is going to take less time. We're going to go from the 2-cell stage into the 4-cell stage, the 4-cell stage into the 8-cell stage, the 8-cell stage into the 16-cell stage, and the 16-cell stage into the 32-cell morula. And this is going to be right here around 3 or 4 days after fertilization. Now, the morula is going to make its way out into the uterine cavity, where it's going to be exposed to the secretions of the endometrium. The name of these secretions are referred to as uterine milk, and they're going to flow into the morula and cause it to rearrange itself into the blastocyst. And then for about 2 days, this blastocyst is going to hang out inside of the uterine cavity and eventually implant into the wall of the endometrium. This implantation takes place right around 6 days. So it is at this point that we're going to see the endometrium accepting the blastocyst and development is going to take off. Here you can see the edge of the endometrium and the developing embryo shown right here. So this picture that we're seeing here is occurring about 1 week after implantation has taken place. Now, inside of the endometrium, there's going to be these spaces known as lacunae. And as the embryo continues to make its way into the endometrium, it's going to erode this tissue and eventually link up the spaces that are the lacunae. This allows blood to flow into the lacunae, which will be the prime means that the embryo gets all of its nutrients and oxygen from the mother. Now, right here towards the end of the second week, right around 13-14 days, we're going to start forming the chorionic villi. Those are going to be projections here from the layers of the trophoblast that are going to bring the fetal blood supply in very close proximity to the mother's blood supply. You can see on this picture more developed chorionic villi with the blood supply of the fetus shown right here. The chorionic villi are going to grow outwards, and then the blood vessels are going to grow into the chorionic villi, which allow for very close proximity between the fetal blood supply and the mother's blood. Now, one thing for us to keep in mind that's very, very important is that the mother's blood supply is not directly connected to the fetal blood supply. All oxygen and carbon dioxide transport takes place by diffusion across these cells. These vessels will have started forming in the connecting stalk, and that's going to give rise to the umbilical cord that's containing the umbilical vein and the umbilical arteries. And this depiction is showing us here right about at 21 days. So at this point, we're about at the 3rd week of development. But another event that takes place in the 3rd week, and really I should say the most important event that's going to take place during the 3rd week, is a process known as gastrulation. Gastrulation is the conversion of the bilaminar embryonic disc into a trilaminar embryonic disc that's going to show us the 3 embryonic germ layers. Before gastrulation has taken place, the embryoblast will have developed into an epiblast and a hypoblast, and they're going to form this tiny little disc, and that is our bilaminar disc. There's 2 layers, hence it's bilaminar. But we're going to take that bilaminar disc and convert it into a trilaminar disc, including the ectoderm, mesoderm, and endoderm. Now, it's important for us to go over the endoderm, mesoderm, and the ectoderm because all of our different tissues are going to arise from these 3 germ layers. Now, there's quite a bit of information that you can see on this slide, so I hope that after going through this with you, you'll realize that the information presented here is much more straightforward than you think. Let's take a look at a few of the structures that arise from the endoderm. So the first thing we see is the epithelial lining of the gastrointestinal tract, the oral cavity, as well as part of the epithelial tissue of its glands. We also have the epithelial lining of some other digestive organs, including the gallbladder and the liver, as well as the urinary bladder. That's not a digestive organ, that will be part of the urinary system. We also will find the epithelial lining of the structures in the respiratory system, and also much of the epithelium of our endocrine organs. So as I've gone through this, hopefully you can see the trend. What we will find with the endoderm is that it's going to give rise to our body's epithelial tissue. In general, epithelial tissue comes from the endoderm. Now, there are a few exceptions to this rule, but on the test, I'm not interested in giving you trick questions about where specialized types of epithelial tissue come from. What you need to understand is that the endoderm is giving rise to our epithelial tissue. Now, let's take a look at the mesoderm here. The skeletal muscles, cardiac muscles, as well as most of our smooth muscles are going to be coming from the mesoderm. That's about all the muscle tissue that we have in our body, so we can say comfortably that muscle tissue in general comes from the mesoderm. But we've got a couple other tissues in here as well, including cartilage, bone, and some other connective tissues, including blood as well as lymphatic tissue. All of these structures are connective tissues. So what we're seeing here is that our four principal types of tissues are coming from these different germ layers. Muscular tissue and connective tissue are coming from the mesoderm, and epithelial tissue is coming primarily from the endoderm. Now that leaves us with one type of tissue, and that is the nervous tissue. Nervous tissue is going to arise from the ectoderm. Now one thing that I will specify here about the ectoderm is something that I said earlier. Everything is not so cut and clear as I'm making it out to be. On the test, it will be pretty cut and clear, but in reality, there is in fact a little bit of overlap here. One thing that you'll see is the erector pili muscle is coming from our ectoderm as well. That is in fact a muscle, but that is also coming from the ectoderm. Now one of the other things that we see that's coming from the ectoderm here are the hair follicles, the nails of our bodies, as well as the epithelial tissue of our skin glands. Now even though we have a lot of exceptions right here, something that you might have noticed about the structures that are shown right here is that these are all the structures that make up the integumentary system. So what we can comfortably say is that epithelial tissue comes from the endoderm, muscle tissue and connective tissue comes from the mesoderm, and the nervous tissue, with the exception of the integumentary system, will come from the ectoderm. We'll get most of the tissue for our special sense organs from here as well, but all I want you to keep in mind is that the ectoderm is giving rise to the nervous tissue as well as the integumentary system. Now beginning in this embryonic period, placentation is going to start taking place. Placentation is the formation of the placenta, and the placenta is the entity that's going to facilitate nutrient and oxygen transport into the fetus. Waste products will also exchange between the mother and the fetus through the placenta as well, and we'll come to see a little later on in this presentation that the placenta is going to be releasing certain hormones that will also help in the development of the fetus and also important for sustaining the pregnancy. You can actually see here that the placenta is going to have two different layers to it, a fetal portion as well as a maternal portion. Now just a disclaimer, this picture is showing you this at the 12th week, which would technically be in the fetal period at this time. I don't plan to make a big distinction about this on your test. I more so am concerned that you guys understand the anatomy of the placenta. The fetal portion here is going to be made up of the chorionic villi. You can see the umbilical cord coming out here, and the chorionic villi that have grown outwards and are housing the blood vessels of the fetus. Now while these chorionic villi were forming, they were eroding into the wall of the endometrium. They were breaking down the tissue and allowing the lacunae to form what are known as intervillous spaces. These intervillous spaces are going to fill up with the maternal blood, and then we're going to have nutrient and waste exchange between the mother's blood and the fetus's blood. Eventually, when fully developed, the placenta is going to sort of look like a pancake. It's going to have the umbilical cord coming off right here, which will contain the umbilical arteries and the umbilical vein. You can see the placenta shown right here. Now we've just talked about the process of gastrulation, which takes place during the 3rd week of development. We're moving on here to the 4th week, and we're going to have a very, very important process that's taking place right now. That is known as embryonic folding. During gastrulation, we formed a trilaminar disc with the ectoderm, the mesoderm, and the endoderm. But here in the 4th week with embryonic folding, we're going to find that the embryo is going to undergo folding in two different directions. The sides of the embryo are going to fold downwards, and you can see that in a transverse section right here, and also the front and the back are going to fold down as well. So this is actually going to form a cylindrical structure here during the 4th week. Now, embryonic folding is not the only process that takes place in the 4th week. We're going to see that organogenesis is going to begin in the 4th week. We're going to see that the upper and lower limbs are also going to start developing here in the 4th week, as well as the heart prominence. And also, the embryo is going to have a tail at this point as well, which will eventually disappear. Now, organogenesis is the development of our major organ system. From the 4th week through the 8th week, organogenesis is going to take place. And by the 8th week, all of the major organ systems will at least be present in some regard. Things are still going to have a ways to develop at that point, but what we're going to find is that as we enter the fetal period, very few new structures are going to show up. Most new organ systems and body structures are going to show up here in the embryonic period. And we're going to have very, very quick development of the structures that are showing up at this time. Here during the 5th week, we're going to see that we have rapid development of the brain and the head. And while we do see the limbs developing during this period, their distinction will be here during the 7th week. The limbs are going to become much more distinct, and we're also going to start seeing the digits arising on the limbs as well. It is here at the 8th week that the embryonic period is going to come to a close. You'll be able to see the eyelids, distinguish the sex based on the external genitalias that are starting to differentiate at this point, and the tail will be disappearing by the 8th week as well. Not only are the upper and lower limbs distinct, but the digits are going to be distinct also. So it is the 8th week that will mark the end of the embryonic period. Beyond this, we're going to have the fetal period that's going to begin at week number 9 and stretch all the way until the 9th month. As I mentioned earlier, very few new structures are arriving during the fetal period. Most of the structures that we're going to see are present at this point. But what we will see with the fetal period is that this is a time for rapid growth, differentiation, and development of the structures that are already present. You'll see some of the events that are taking place at different times during the fetal period. I know there's a lot of information on these slides, so really give some focus to what I've highlighted in red here. One thing that we'll see here between the 9th and the 12th week is that the heartbeat can be detected, and we're also going to be able to distinguish the sex of the baby. The external genitalia will have started to differentiate at this point. Between weeks 13 and week 16, the eyes are going to move a little bit more medially, and the ears are going to move a little bit laterally towards the sides of the head. The lower limbs are lengthening during this period, and rapid development is taking place throughout the entirety of the fetal period as well. Here between week 17 and week 20, head hair and eyebrows are going to be visible, and the mother will often feel the movements of the fetus as well. Now at this point, development is taking place rapidly. However, babies that undergo a premature birth at this stage will not survive. It is only here between week 21 and week 25 that we see fetuses surviving if born prematurely. A fetus at 24 weeks of age can survive if it is born prematurely. And the reason for this is by this point the respiratory system is just about finished developing. Here you can see that between weeks 26 and weeks 29, because the lungs can provide adequate ventilation and the central nervous system is developed, this allows the baby to control its own breathing and also to control its own body temperature. These are very vital things that a fetus is going to need to survive outside of the womb. Now also during this period, we're going to see the toenails showing up as well as the fingernails. There's going to start to be a deposition of body fat on the fetus, and this is also the stage where the testes begin to descend. A few weeks after this, we'll see that body fat is going to start increasing and even further depositing as this process continues. Now one thing that we definitely need to keep in mind is that even after birth, infants are not completely developed. Development is going to take place throughout the entirety of life. And provided that the baby is given everything that it needs, development should happen of its own accord. So I know that there's a lot of information about the fetal period, but again, as I mentioned, focus on what's highlighted in red. Starting off at fertilization, implantation took about 6 days, and then the embryonic period is going to stretch from implantation all the way to the end of week number 8. Week number 9 is going to start off, and we're then going to start calling the baby a fetus. As long as that fetus makes it to about 25 weeks, likelihood of its survival has significantly gone up. And the entire process of gestation should take about 38 weeks. Now, as we're talking about pregnancy and development, it's very important that we mention teratogens. Teratogens are any type of chemical agent or chemical substance that can lead to a developmental defect in an embryo or a fetus. There are many different types of teratogens out there, but one of the most common ones is alcohol. Alcohol can lead to fetal alcohol syndrome, which is the #1 preventable cause of birth defects. We'll find that teratogens can be viruses, different types of chemicals, excess hormones, and antibiotics, and to be even more specific, cocaine. All of these substances can cause birth defects if allowed to access the fetus during development. One other very serious teratogen to be taken into account is cigarette smoking. Higher mortality rates, low birth weight, and anencephaly have all been linked to cigarette smoking during pregnancy. In case you were not aware, anencephaly is the absence of the brain. So lots of very bad birth defects can be linked to cigarette smoking. Now the last teratogen that I'm going to mention is ionizing radiation. Now this sounds pretty severe. Generally, we don't go about our days thinking that we're coming in contact with ionizing radiation. However, one source of radiation that we need to be careful of are X-rays. Radiation can lead to microcephaly, mental retardation, and other defects with the skeletal system. It is for this reason that women are often asked if they are pregnant prior to taking an X-ray. There are quite a few teratogens out there. However, by following the advice of medical professionals, combined with discipline, birth defects due to teratogens can be significantly decreased.